Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is for 29th December 2020. As we have been training ourselves in the pattern of assertion and reasoning questions, today in this session we are going to answer some of the assertion and reasoning questions from chapter number 2, sexual reproduction in flowering plants. And as we have been training for the last 15-16 days, you know the directions, and the directions are: answer A if both assertion and reason are true and reason is the correct explanation of assertion. Answer B if both assertion and reason are true but reason is not the correct explanation of assertion. Answer C if assertion is true but reason is false. Answer D if both assertion and reason are false. So with that background in mind, let's move ahead. Question number 1. Assertion. An angiospermous flower represents the modified condensed shoot which performs the function of sexual reproduction. I think the statement is correct. Yes. Reason. The fertile leaves of the shoot become modified into microsporophylls and megasporophylls which bear ovules and anthers respectively. I think this is false. Reason is false. So if assertion is right and reason is false, the answer has to be C. Why is it so? Why question number 1 is C? For that, what we need to do is that why question number 1 C is a flower is a modified condensed shoot specialized to carry out the function of sexual reproduction in angiosperms. Like a branch, it arises in the axil of small leaf-like structure called bract. The receptacle, that is thalamus, of a flower supports all the floral appendages like sepals, petals, stamens and carpels. The receptacle consists of several crowded nodes which are separated by condensed internodes. The fertile leaves of shoot become microsporophylls, stamens, and megasporophylls, that is carpels, which bear anthers and ovules respectively. The anthers produce pollen grains and the ovules possess eggs. That is the reason why question number 1 is C. Now question number 2, assertion. A typical microsporangium of angiosperms is generally surrounded by 4 wall layers, that is epidermis, endothecium, middle layers and tapetum. Reason: the outer 3 wall layers perform the function of protection and help in dehiscence of anther to release the pollen. I think assertion is correct, reason is correct, but reason is not explaining the assertion, so the answer has to be B for question number 2. And now question number 3, assertion. In a microsporangium, the tapetal cells possess little cytoplasm and generally have a single prominent nucleus. Now the reason. During microsporogenesis, the microspore mother cells, that is MMCs, undergo mitotic divisions to produce haploid microspore tetrads. I think both the statements are wrong, assertion and reason. The answer has to be D. And why D? Tapetum is the innermost wall layer of a microsporangium. It nourishes the developing pollen grains. The tapetal cells enlarge rapidly and become filled with dense protoplasmic contents as well as nutrients. Tapetal cells are generally multinucleate or their nucleus becomes polyploid due to endoploidy. Microsporogenesis refers to the process of formation of haploid microspores or pollen grains from a microspore mother cell MMC or PMC pollen mother cell through meiosis. Now through meiosis, whereas here we are saying mitosis. That is the thing. And so the answer has to be D. Question number 4. Assertion. In most angiosperms, microspores of a tetrad grow and separate from one another shortly after meiosis. I think correct statement is correct. Reason. In the members of families of Achidaceae and Asclepiadaceae, all the pollen grains of a sporangium remain united to form a compact structure called polonium. I think the assertion as well as reason correct statement, but reason is not explaining the assertion. Hence the answer should be B. Because we are talking here, it is microspores of tetrad. In assertion, in reason we are talking something else of these. That's why. Question number 5. Assertion. Exine, that is outer covering, of pollen grain is made up of sporopollenin, which is resistant to high temperature, strong acid or alkali as well as enzymatic degradation. Correct statement. Recollect from your memory. We were talking about this sporopollenin and I said this is important question. And then reason, sporopollenin is absent in the region of germ pores. I think again the reason is not explaining the assertion, so it has to be B. Question number 6. Assertion. The method of development of embryo sac from a single functional megaspore is termed as monosporic development. Reason. In monosporic, that is polygonium type of embryo sac development, usually the megaspore, which is situated towards micropolar end, remains functional. I think again assertion is correct and reason is wrong. So answer has to be C. Why C? In monosporic, that is polygonium type of development of embryo sac, only one megaspore situated towards chalagal end remains functional. Chalagal end. The remaining 3 megaspores gradually degenerate and finally disappear. The functional haploid megaspore enlarges in size and by means of 3 successive mitotic effusions gives rise to an 8 nucleate embryo sac. This type of embryo sac development occurs in majority of the flowering plants and the common example is polygonum. So that is the reason why answer is C. Question number 7. Moving ahead. Although zetonogamy is functionally cross-pollination involving a pollinating agent, genetically it is similar to autogamy. Correct. Reason. In zetonogamy, pollen grains from the anthers of one flower are transferred to the stigma of another flower born on the same plant. So both assertion correct, reason correct, explanation okay. Hence answer to be A. Zetonogamy is a type of pollination in which pollen grains of one flower are transferred to the stigma of another flower belonging to either the same plant or genetically similar plant. It usually occurs in plants which show monoecious condition, that is unisexual male and female flowers are born on the same plant. Thus zetonogamy is functionally cross-pollination as it involves the pollinating agent to carry out pollination. But genetically speaking, it is similar to autogamy, that is almost self-pollination, since the pollen grains come from the genetically same plant. Question number 8. Assertion. Hydrophily, hydros, water. Phily, love for water. Hydrophily constitutes a major mode of pollination in most of the aquatic angiospermous plants. Reason. Almost all the aquatic dicot and monocot plants require water for the transport of male gametes and for fertilization. I think assertion wrong, reason wrong. The answer has to be D. So YD, hydrophily is the mode of pollination, that is transfer of pollen grains from mature anther of a flower to the stigma of another flower, which is accomplished through the agency of water. Pollination by water is quite rare in flowering plants and is limited to only about 30 genera, mostly monocotyledons, that is Vallisneria, Zostera, Ceratophyllum, etc. In many aquatic plants with emergent flowers, pollination occurs by wind or insects like lotus, water lily, water hyacinth, etc. Hence, answer has to be D. Question number 9. Assertion. Only the pre-pollination growth of male gametophyte occurs inside the microsporangium, whereas the remaining growth occurs over the female reproductive organs. Okay, region. Whole of the growth of female gametophyte occurs inside the megasporangium. I think both the assertion reason correct, but reason is not explaining the assertion, hence B. Question number 10. Assertion. Self-incompatibility is a genetic mechanism which prevents self-pollination and thereby fertilization by inhibiting either pollen germination or pollen tube growth in the pistil. Reason. In gametophytic self-incompatibility, the incompatibility reaction is determined by the genotype of the sporophytic tissue of the plant from which the pollen is derived. So, question number 10 has to be C. Why C? Self-incompatibility refers to the inability of pollen from a flower to germinate on the stigma of the same flower or other flower of the same plant. Depending on the origin of factors determining the mating types of two categories of self-incompatibility have been recognized. One is gametophytic self-incompatibility, we say GSI. The incompatibility process is determined by the genotype of male gametophyte, that is pollen itself. Example, Liliaceae, Poaceae, Solanaceae. And the second one, sporophytic self-incompatibility, SSI. The incompatibility process is controlled by the genotype of sporophytic tissue of the plant from which the pollen is derived. Example, Brassicaceae, Asteraceae. So, hence, the answer is C. Question number 11. Assertion. In angiosperms, endosperm development precedes embryo development. Correct. Reason. The double fertilization ensures that the nutritive tissue is formed before the zygote starts cleaving, so that energy spent on the formation of endosperm does not get wasted. I think perfect A. Yes. During double fertilization in angiosperms, one male gamete fuses with the egg to form a diploid zygote. We call it syngamy or generative fertilization. The diploid zygote finally develops into embryo. The other male gamete fuses with the two polar nuclei or secondary nucleus to form a triploid or triploid primary endosperm nucleus. We say PEN. PEN, that is primary endosperm nucleus, resulting from triple fusion or vegetative fertilization. Double fertilization ensures that the nutritive tissue is formed before the zygote starts cleaving, so that the energy spent on the formation of endosperm does not go waste in case fertilization fails. Angiosperms are therefore economical and more specialized as compared to gymnosperms, where a large nutritive female gametophyte is formed long before fertilization. Hence, answer A. Question number 12. Assertion. In Cocos nucifera, coconut water represents the cellular endosperm, and the surrounding white kernel represents the free nuclear endosperm. I think wrong. Reason: endosperm is always completely consumed by developing embryo before seed formation. I think both the statements are wrong. Answer: in Cocos nucifera, that's coconut, the coconut water represents a free nuclear endosperm, and the surrounding kernel represents the cellular endosperm. Endosperm may either be completely consumed by the developing embryo, like in pea, groundnut, beans, before seed maturation, or it may persist in mature seeds like castor, coconut, and is used during seed germination. Hence, answer has to be D. And now I need to go to the next page, question number 13. That is, assertion. During embryo development in dicots, suspensor serves as the main nutritive tissue for the embryo. Reason: the last cell of the suspensor at the end adjacent to the embryo is known as haustorium. I think both the statements are wrong. D. During embryo development in dicots, the suspensor cell divides transversely a few times to produce a filamentous suspensor of 6 to 10 cells. The first cell of suspensor towards the micropylar end becomes swollen and functions as haustorium, whereas the last cell of suspensor at the end adjacent to the embryo is known as hypophysis. The function of the suspensor is to anchor the embryo to the embryo sac and push it deep into the endosperm so that the embryo lies in a nutritionally favorable environment. Now, question number 14, assertion. X albuminous seeds do not possess any residual endosperm as it is completely consumed during embryo development. Reason: wheat, castor, pea, and groundnut are all the examples of X albuminous seeds. I think this is wrong. So it has to be C. In some seeds, endosperm persists in the seed as food storage tissue. Such seeds are called endospermic or albuminous. Example: castor, maize, wheat, barley, rubber, coconut. In others, the endosperm is completely eaten up by growing embryo. The food for later development of embryo is then stored in cotyledons, which becomes massive. Such seeds are non-endospermic or X-albuminous. Example: pea, gram, beans, and groundnuts. Now, question number 15. Assertion. In plants, apomixis is a form of asexual reproduction that mimics sexual reproduction. Correct. Reason: apomixis involves the production of seeds without the fusion of gametes. I think it has to be. Apo means without, mixis means fusion. Without fusion, apomixis is a mode of reproduction in some plants that results in the formation of seeds without gametic fusion. It is a form of asexual reproduction. In plants, apomixis commonly mimics sexual reproduction but produces seeds without fertilization. Some species of Asteraceae and grasses, there are several methods of apomictic development in seeds. Hence, for question number 15, answer is A. So this is how we need to arrive at the answers with the concept at our back of our mind. So with this, I have completed the entire syllabus of biology, all chapters, because first chapter is not there for your batch.